Well, welcome to the fine structure of myelin. I'm Dr. Rushmore, and our goal today is to see what myelin looks like in the EM and to identify the parts that are discernible in the EM. So as a point of reminder, this is a drawing of a neuron by, yes, yours truly. And a neuron consists of a cell body marked with an X, processes that come out and ramify locally, which are called dendrites, and then one singular process which comes out and extends for a long distance from the cell body before ending in branches and in terminal boutons. Now we're interested in what happens before the terminal boutons and after it leaves the cell body. And if we do so, we find that right initially after leaving the cell body, it acquires a particular appendage, and this is a sheath. It's very much like the installation on plumbing in this sort of drawing. So the things that we can get from this drawing is that these installation sheets are segmental, they're individual. And if we look down here, we can see that on one of these segments of insulation or myelin, we see that there is a cell body attached. And so this appendage, this myelin, is associated with particular cells and produced by particular cells. In the central nervous system, myelin is produced by a cell called the oligodendrocyte. And in the peripheral nervous system, it is produced by a cell called the Schwann cell. Alright, so let's take a higher power look at a cartoon myelinated axon, shall we? And when we do so, we see something along these lines, where green is the axon, and the sheath is here in gray. So where the axon is naked is called the node of Ranvier. And we see three nodes in this cartoon image. As a result, we call this an internode of myelin. And at least in the periphery, one Schwann cell produces one internode of myelin. So now what we're going to do is see what happens when we cut this in transverse section and look at it at very high power on the EM. This is sort of what we see. Alright, so let me point out a few things. The first thing to point out is that this is the axon, and I can draw the border of the axon plasma membrane like this. That means that immediately surrounding the axon is myelin. And this very stereotypical dark cylindrical almost laminated structure is the myelin. And we see myelin here, here, and here, and here. So what is everything else? Well, I told you that myelin occurs both segmentally, it occurs in units, and it is created by cell. And in this case, we're in the peripheral nervous system, and so we can identify the cell in this particular EM. This is the nucleus of the Schwann cell. And we can draw the border of the Schwann cell, the plasma membrane, like this. And this delimits the Schwann cell and allows us to make an important observation. That is to say that the axon, which is a process of a cell that is somewhat distant, is totally encapsulated by the Schwann cell, and by the Schwann cell process, the specialization that makes myelin. Okay, so let's take a closer look at what myelin is per se. When we take a higher view, we see the same thing. We see that this is the axon, here's the Schwann cell nucleus, and this is myelin. And at higher power we can see an alternating series of light and dark lines. Okay, we're going to go in higher power even more. Let's see that button. And this is what we see here. This is the inside of the axon. And this is going to be the Schwann cell cytoplasm. And at this high power, which is insanely high power, we can see that the the myelin sheath is comprised of thick discontinuous lines, as seen here in green. And there's one right here. And in fact, it's every other line that you see. And we see another qualitatively different, sort of lighter line, or less dense line, illustrated here in blue. And so our myelin sheath appears to be made up of two alternating series of lines. And the blue line is known as the intraperiod line. And the green, yes, the green is known as the major dense line. And the major dense line is, of course, majorly dense and forms a particular periodicity, and that periodicity is broken up by the intraperiod line. So the dense one is the major dense line, the less dense one is the intraperiod line. So now our goal is to figure out how this comes about. So it's actually fairly straightforward, but requires a few steps to think about. So first thing we're going to do is draw our axon. And then we're going to draw our Schwann cell, which is in fact going to wrap a process around our axon. Something like this. So this is our Schwann cell, and this is our axon, and we see a process. Now I'm going to tell you that what we saw at the myelin here, the myelin is simply a process of the Schwann cell that has been wrapped around and around and around the axon. And so we're going to now figure out and try to interpret why you have a major dense line and why you have an intraperiod line. In other words, why you have a heterogeneity of sort of electron microscopy lines in the myelin itself. So what we're going to simply do is we're going to take a window like this and deconstruct it. So we have 1, 2, 3, and 4. This is 1, this is 2, this is 3, and this is 4. That means that this is the cytoplasm of the Schwann cell. And so we're just going to denote that like so. And that means that this space here, X, is this space here, and this corresponds to extracellular space. So we have plasma membrane, cytoplasm, plasma membrane, cytoplasm, plasma membrane. Okay, but since we are budding histologists, we know that the plasma membrane is just not one line, it is two lines. Okay. It's two lines because this line represents the phospholipid heads on one side, and this line represents the phospholipid heads of the bilayer on another side. So this line is that, this line is this, and the space between the two lines represents the space where the tails are. And these do not get labeled or stained by the metals in EM. And so a particular plasma membrane at high enough magnification is actually two lines, and is referred to as the unit membrane. Okay, so let's add those in. Let's add in this more appropriate idea. So plasma membrane 1, plasma membrane 2, plasma membrane 1, plasma membrane 2. The hatched mark indicate cytoplasm, and the X is extracellular. Okay, so the first step to understanding this is that there are two lines that make up each plasma membrane. The second step, and there are only three steps, the second step is that the plasma membrane in contact with the cytoplasm is associated with proteins, and so that makes it denser. So if we know that the cytoplasmic leaflet of the plasma membrane is denser than the extracellular leaflet, then we can redraw this more appropriately, something like this. Okay, we'll crosshatch the presence of cytoplasm, and X marks the extracellular space. Okay, so the next thing is the last thing to remember, and that is that the cytoplasm and the extracellular space get sucked out until this gets opposed to this, and this gets opposed to this. So all the space gets sucked out, so all you're left with are plasma membrane. So let's start with sucking out the cytoplasm, and we'll do that up here. When we do that, we eliminate the crosshatch area. We have the plasma cell leaflet that's in contact with the extracellular matrix, and the extracellular fluid rather, and then we have two reasonably dense lines that represent the phospholipid heads and the proteins here and here. So this has just come together with this to produce this. We then have another line which represents the extracellular leaflet. We have extracellular space and the same thing happening on the other side. So this is the joined intracellular leaflet from one side and then the other side once we suck out the cytoplasm. Okay, so now we're going to suck out the extracellular fluid. And when we do that, we have one line, a very thick line, another line, and this line come together to do this, thick line, and a thin line. And if we keep going, we wrap our process around process, we have now an alternating series of thick and thin lines. So this is all to say that this guy, which are the opposed cytoplasmic surfaces of the Schwann cell, create the thicker line, which is our major dense line. And this guy right here, which are the opposed extracellular leaflets, equals the intraperiod line. And so this is how we interpret those two lines. This is how we interpret the myelin sheath. Here's that picture again. We have the axon, we have the Schwann cell cytoplasm. We have these lines marked in green. These are the major dense lines, which correspond to the fused or opposed cytoplasmic leaflets of the Schwann cell plasma membrane. And here in blue is the intraperiod line, which is the fused or opposed extracellular leaflets of the Schwann cell plasma membrane. Okay, in general, we can think about it very similar to a roll of toothpaste. This is our axon, and this is the Schwann cell. It first rolls around the axon and then squeezes out all the cytoplasm and squeezes out all the space to form this very regular array. And this process is our myelin. And these are the two major points that I would like you to remember. The major dense line is the opposed cytoplasmic leaflets, and the extracellular leaflets makes up the intraperiod line. So that's all for today. Thank you, and see you next time.